Jean-Pierre Changeux is a French neuroscientist known for his research in several fields of biology, from the structure and function of proteins, to the early development of the nervous system up to cognitive functions. Although being famous in biological sciences for the MWC model, the identification and purification of the nicotinic acetylcholine receptor, and the theory of epigenesis by synapse selection are also notable scientific achievements. Changeux is known by the non-scientific public for his ideas regarding the connection between mind and physical brain. As put forth in his book, Conversations on Mind, Matter, and Mathematics, Changeux strongly supports the view that the nervous system functions in a projective, rather than reactive style, and that interaction with the environment, rather than being instructive, results in the selection amongst a diversity of pre-existing internal representations. Changeux was born in Daumont, France. He entered the École Normale Supérieure de Copyright in 1955, where he obtained a bachelor's degree in 1957 and a master's degree in 1958. He also received his AGR École de Copyright in natural science the same year. He began his scientific career during his ENS years during summer internships in Banyuls-sur-Mer, where he identified a new genus of parasitic copepod. He pursued PhD studies at the Pasteur Institute under the direction of Jacques Monod and François Jacob, and gained his doctorate in 1964. Changeux then left France for postdoctoral studies, first at the University of California, Berkeley, then at Columbia University College of Physicians and Surgeons, New York. He returned to France's ATTACH École de Copyright to the chair of molecular biology held by Jacques Monod. In 1972, he became director of the unit of molecular neurobiology at the Pasteur Institute, where he received a professorship in 1975. In 1975, Changeux was elected professor at the École d'Aérosciences de France, chair of cell communications, position that he held until 2006. Changeux is author of more than 600 scientific articles and several books, technical or for general audience. All his scientific career, Changeux has been faithful to a handful of scientific questions, at molecular, cellular and brain levels. If one needs to seek a unifying theme to all of them, it is the conviction that selection is the basis of life processes, rather than instruction. While started as separate lines of investigations, all the research threads were tied in the recent decades within the study of allosteric mechanisms as a basis for the involvement of nicotinic receptors in cognitive functions. During his PhD studies in the laboratory of Jacques Monod and François Jacob, Changeux studied the allosteric regulations of enzymes, that is, the modulation of their activity by compounds different from their substrates. This work led to the development of the model of concerted transitions for allosteric proteins. The main ideas behind this theory are: 1. Proteins can exist under various conformations in thermal equilibrium in the absence of regulators. The allosteric regulators merely shift the equilibrium between the conformations, stabilizing the ones for which they display the highest affinity, and to all the subunits of a symmetrical multimeric protein exist in the same conformation, the transition taking place in a concerted fashion. The resulting model explains the observed cooperativity without a progressive change of biophysical parameters. This conceptual framework is still the principal model used to explain the function of cooperative proteins such as hemoglobin. In his PhD thesis, Changeux suggested that the recognition and transmission of signals by membrane, and in particular by synapses, could use the same mechanisms as the allosteric regulation of enzymes. More than 40 years of research would follow, mainly focused on nicotinic acetylcholine receptors. In 1967, Changeux extended the MWC model to bidimensional lattice of receptors. He then applied this idea to the postsynaptic membrane of electric organs. His team demonstrated the existence of several interconvertible states for the nicotinic receptor: resting, open and desensitized, displaying different affinities for the ligands, such as the endogenous agonist acetylcholine. The transitions between the states followed different kinetics, and those kinetics plus the differential affinities suffice to explain the shape of the postsynaptic potential. A full mechanistic model of the nicotinic receptor from striated muscle was to be provided much later, when Changeux collaborated with Stuart Edelstein, another specialist of allostery, who worked decades on hemoglobin. In addition to the allosteric modulation of the channel gating by the agonists, many other regulations of the ligand-gated ion channel's activity have since been discovered. The modulators bind to a variety of allosteric sites, whether on the agonist binding sites, other binding sites at the subunit interfaces, on the cytoplasmic part of the protein, or in the transmembrane domain.